Next, we're going to talk about DNA replication. First, DNA replication is known as semiconservative. What this means is that we have an original strand of DNA, and I'm drawing this DNA kind of as a ladder on its side. And what happens is the strands of DNA split and new DNA is made on the insides using the old DNA as a template, so that each replicated strand of DNA is half old and half new. And that's why we call it semi-conservative. So that's what our DNA ends up looking like. There are several steps in the process and several enzymes that mediate DNA replication. To begin, we start with helicase. So if we draw our strands of DNA again, so in step 1, helicase unzips DNA to open up and separate those strands. So we go in here, helicase is basically going to come in here and open up the strands of DNA. It's important to mention that DNA is, again, anti-parallel, meaning that the strands run in opposite directions. So we have 3', 5', 5', 3'. And DNA can only be created in the 5' to 3' direction. So our strands have an issue. One strand is opening up in this direction, and we can lay down new DNA, 5' to 3'. The other strand, though, is running in the opposite direction. So as helicase is traveling in this direction, we actually have to synthesize DNA backwards because it can only be made as a 5' to 3' direction. So as helicase separates the strands, we end up with two different types of scenarios. A leading strand, which travels in the same direction as helicase and can be synthesized continuously. And a lagging strand. And this is going to be made in the opposite direction as helicase. And it's going to have to be made in chunks. So as one section opens up, this can be replicated. Now the next section is going to open and it's going to be made backwards in chunks. And those chunks are called Okazaki fragments. So we're going to continue on with the next step. And erase this stuff. Okay. So step number 2. DNA can't just be made de novo. De novo means like from nothing, basically, from scratch. We have to have a primer before we can start laying down new DNA. And this primer is made up of RNA. So we have this enzyme called primase, which lays down an RNA primer. So the primer is going to be laid down here, but for the lagging strand, it's going to be laid down here because remember, we have to make DNA backwards. Now, eukaryotic replication is a little bit more complicated than prokaryotic, so we're going to focus on kind of the simpler process of prokaryotic replication. And with prokaryotic replication, in the next step, we have DNA polymerase 3. And DNA polymerase 3 is going to begin synthesizing new DNA. So from this RNA primer, DNA polymerase is going to start adding new bases. From this primer, DNA polymerase is going to start adding new bases. So continuing on with the next step, DNA helicase is going to keep opening up these strands. And the leading strand is going to be made continuously because it's going to follow the direction that helicase travels. With the lagging strand, we laid down an RNA primer and we synthesized DNA backwards. Then we had to lay down another RNA primer when the next section opens up, and we again synthesized DNA backwards. Then we lay down another RNA primer, and you kind of get the point. Keeps being synthesized backwards in these chunks called Okazaki fragments. As helicase opens up the next section, the top strand will be able to just be continuously synthesized, but the bottom strand is going to have to wait until we have another primer laid down so that it can be synthesized backwards. Now this is a problem. First of all, if this is our completely replicated DNA strand, we have these RNA primers, and we can't have any RNA inside of the DNA. We also have these gaps between the primer and the next segment of DNA. So, to fix this, we have RNA primase, DNA polymerase 1. Which is going to correct any errors. So if we had an A here and we ended up putting in a C over here, it would fix that for us. And it's also going to remove these pieces of RNA and replace them with DNA. So we come in there, cut out the pieces of RNA, and we replace them with DNA. But we have one last problem. Like I kind of mentioned, we have these gaps that exist between the strands. So the last step, ligase, is going to glue together those Okazaki fragments. So what we end up with is what we were hoping for. We have two strands, so it's replicated. Half of it's old, half of it's new. Now it's important to mention the purpose of DNA replication. Why do we have to make a copy of all of our DNA? Well, let's say you get a paper cut and you've damaged a bunch of cells in your skin. Those cells that are dead have to be replaced. In order to replace those dead cells, we're going to have to make new cells. These new cells are going to need a full copy of DNA. Otherwise, they won't be able to make proteins or do their normal function. So we have to replicate our DNA in order to allow for cell division so that we can make more copies of our cells.